shear force that goes through your tissues. But I also think about when it comes right down to basically the hair cells in terms of basically, so for your hearing. That's a force that's sent at a very, 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 much different length scale than actually the force loads that are put on your bones. And basically what I'll do is talk about what goes wrong when those forces are going wrong. So as I just mentioned, you can have different types of stresses or forces that tissues and cells will experience. So shear, like blood flow going through your system. The endothelial cells that are in your blood vessels, in your vasculature, they're experiencing a shear flow. So shear stress is stress applied parallel to the surface of the cell. So it's coming along, basically through your blood vessels. That's a shear force. Okay. Compression is stress applied perpendicular to the cell, and it results in compaction. So I was talking about you're putting load on your bones. So basically that's a compression loading. And tension is a stress that's applied perpendicular to the cell that leads to expansion. Okay, so you've got sort of three types of forces that you're experiencing. What I put this in here is to mention that if you just go back early embryogenesis, just at the stage where basically the egg is fertilized, and you look at things like actin reorganization, relocalization, and myosins, you already, even at that stage, begin to have changes in mechanical properties of the cell. So it basically goes right back to early embryogenesis, and this is some work where they're looking at memory deformation, and in this case it's Drosophila, but it's really talking about the fact that cells, the rheology of cells changes. Cells stiffen as you start to develop into an organism. So basically forces are changing as early as that. And another way to think about this is, for example...
What is rheology?
So these guys can... I've got a couple of mechanical engineers here. So basically, I'm going to be discussing kind of what... how do I
rheology is the study of how things flow.
Yeah. Basically, it's kind of like looking at, and I'm getting into later what materials properties are, but it's looking at, sort of in a nutshell, kind of the viscoelastic properties of the cell, right? So it's looking at, and I think engineers and soft particle physicists are still nattering about it because they really can't decide exactly what it is. But it kind of tells you how, actually there's Chris right behind you who does atomic force microscopy to measure cell rheology. But it'll tell you how kind of, I guess the word, it's not just even the squishiness of a cell, but it really gives you a measure almost of the viscoelastic properties of the cell, right, Chris?
Mostly it's stiffness.
Yeah, stiffness. But it's also how basically how dynamic that cell is. So if you have transport processes, proteins moving around, basically, I guess it's everything. It's the flow of living material in that cell as well, right? So it gets a little harder to explain it. I hope it's a little daunting to do this kind of my students.
The Oxford English Dictionary is the study of deformation and flow of matter.
Yeah, but that doesn't seem to be a very satisfying explanation, is it? What it really means is it's kind of how dynamic and how soft and squishy a cell is. That would be. So in other words, what it means is your cells become stiffer as you differentiate into a tissue. So you become stiffer than when you were an egg. I guess that's a very kind of gross way to put it. Another illustration of force, how it regulates this. This is looking at vascular development. So it's really cool because when you start to look at the embryo development, this is kind of the nexus of all the blood vessels. And when the heart starts pumping and the blood begins to flow, now you actually start to make these beautiful, vessels of very different kind of diameters and behaviors. So it's actually coincident with the application or the experience of a shear flow that you have this switch in embryogenesis, which is really kind of a nice illustration. So what I'm going to talk about a little bit today, in a little bit more detail that's relating to cancer, where we're going with this, is that every single tissue in your body has surrounding it an extracellular matrix. And what I like to think about is that we're made up predominantly of water, and actually the reality is we're 20% collagens. Of the proteins in our body, 20% of us, 20 or 30%, actually 30% total is extracellular matrix proteins. Most of us is water. And then the cellular component of that is so minuscule, relatively speaking. So we're mainly extracellular matrix. And if you think about it, Jell-O is collagen. So we're essentially a big ball of Jell-O. and how that exocyte matrix is organized with respect to the cells is really important and really telling. So to ignore the exocyte matrix, to ignore the, what it's doing and how it's actually regulating our behavior is kind of is ignoring the huge component of who we are and what we function like. And so I just put this up here because exocyte matrix is organized in very different types, and it comes in very different biochemical compositions. And each of these exocyte matrix has its own what's called viscoelasticity, which is what I explained earlier, because we are, as a living being, viscoelastic. So our cells, the rheology of our cells is kind of a measure of the viscoelasticity of the cell. Our exocyte matrix, or our tissues, you can measure the viscoelasticity. And one of the ways you do that is you have these unconfined compression devices, which essentially what you're doing is you put a little piece of tissue under here, and you actually exert a force of a known magnitude, and basically you can do it at a certain, velocity. And what you're going to get is a stress-strain curve, and I'll explain what that really means. And it's really, so what you get is a stress-strain, or a measure of the exocyte matrix viscoelasticity. So here's the explanation that I think actually is a little more satisfying for the non-engineering person. So a stress-strain curve is really a measure of the material's properties and how the tissue responds. Okay, so what it really is it's literally a visual readout of the mathematics of how a material responds to a given force. Okay, so strain. So I already told you, what a stress is. Stress is basically over, is like basically force over area. So strain is the inherent material's properties. So how do they respond to that actual force? So if you're really stiff or you're really soft, you're going to have a very different response to an applied force. Okay, and basically the response is going to be proportional to the strain, i.e. the force per given area, and the magnitude of the applied force. And the response of the material to the applied strain is how the material responds. So in other words, does it unfold? Is it really elastic? Is it cross-linked? Is it stiff? So if you took a little piece of your tissue and you basically exposed it, I think I'm doing a reasonable job here. So what you really see is a net stress-strain curve, which is a readout of how sort of stiff that tissue is and how much of that applied force you're putting on it. So what that does is why is it important? Well, I just give a couple of little examples. So basically, in the sort of automotive industry, they're really interested in, trying to design crash helmets and things like that, or understand, how you can, what kind of damage you might get. And one of the ways that you can understand how much damage you might have to an individual is you need to actually understand the geometry of the brain and, of the actual velocity that vehicle is being exposed to. But it also needs to know what the materials properties of the brain. So if you're actually young, you have a softer brain than if you're older. So you're going to respond differently to basically some kind of an injury. So this kind of information is really important. How do you actually study that? Well, there's many different ways that you can study it. One of the really simple ways is that you can actually generate materials that have a very known materials properties or stiffness. In this case, we use things like acrylamide gels, in which we can basically cross-link them and put them on these little cover slips, and then you can conjugate materials of your choice onto them, and then you can put your cells on top, and you can manipulate these cells to actually study what actually the cells do in response to changing materials properties. So if you're a muscle cell and you want to look at myoblast differentiation, for example, what I'd like to show you here is that to get really optimal differentiation of these myoblasts, you actually need to have them in the right stiffness. If it's too soft, they don't differentiate. You see these little striations. If it's too stiff, they also don't differentiate. So there's a sweet spot in terms of differentiation. And if you're a neuron, you like it really soft, and you've got all these nice processes. If you're an astrocyte, so your brain is made of a combination of neurons and astrocytes. If you put the two of these together, basically on a stiffer environment, the astrocytes go crazy, and the neural cells are not able to put out their processes as well. So what this is really demonstrating is that there's a way that the stiffness actually modifies cell differentiation and behavior. And of course, more recently, we and others have started taking this towards stem cell behavior. And we all are recognized that you can, take some of these human ES cells or mouse ES cells out of the blastocyst, and you can start, manipulating the growth factors. And the idea here is that you can then reinsert them into tissues, and then you can use this as a kind of tissue regeneration process. But trying to understand how to control that's been really